Okay, so whilst we are waiting for them to join, I think we can go through what we discussed in our previous meeting. In order for us not to waste time, I think I will just be with you for about an hour and 30 minutes. Then we can end the section for today. So in our previous meeting, or previously on the nervous system, we've been looking at the parts of a neuron and their functions. Is that okay? And I mentioned that for nervous system, you should know everything about a neuron, because the neuron is the basic structural and functional units of the nervous system. So all the functions of the nervous system that we discussed, now those functions, the nervous system are able to perform those functions because of neurons. Okay, and now this is how a typical neuron looks like. Okay, so typically a neuron has a cell body, which we also call the soma. Okay, good. We call a cell body or a soma. Okay, and this is where we have the nucleus of the neuron and several other organelles within the neuron. Okay, apart from the soma and then the structures within the soma, we have the dendrites attached to the cell body or the soma. So these are the dendrites. And I mentioned that the dendrites, if a neuron have to receive an information from another neuron, it receives that signal with the help of the dendrites. Okay, then we have the axon. The axon is the long elongated part. So from this part to this part of the neuron is called the axon. So when we ask you to describe an axon anatomically, an axon is simply the long elongated part of a neuron. So from this point up to this point is the axon. The long elongated part of the neuron is what we call the axon. And this is where impulses are being transmitted. Okay, so the dendrite receives the signal or the impulse, then the impulse are transmitted along the axon through to another neuron. Is that okay? Good. So when we say describe an axon and state the function of an axon is more like the long singular part or projection or the elongated part of a neuron. Is that okay? And then the functions, impulses are transmitted. Impulses that are received with the help of the dendrites of a neuron are transmitted along the axon. Is that okay? Great. Then we also mentioned that we have at the very end of the axon, we have what we call the axon terminals. Okay, we have the axon terminals, or you can call it the synaptic knobs. Synaptic knobs. So these very ones. Okay, so these very ones are the axon terminals. The axon terminals, or you can call them the synaptic knobs. Is that okay? There are some at times that in some textbooks they will call them nerve endings. The nerve endings. And then I also mentioned that we have some neurons that have myelin sheaths. We have some neurons that on the axons they have the myelin sheaths. Is that okay? Some neurons too do not have the myelin sheath. Neurons that have the myelin sheath facilitate fast conduction of impulse. So when we take a neuron with a myelin sheath and a neuron without a myelin sheath, when it comes to impulse transmission, neurons with myelin sheath aid with fast conduction of nerve impulses. Is that okay? That's the difference. And if a neuron is having a myelin sheath, there will be a squam cell within the myelin sheath. Is that okay? Great. So here is it. So we have, this is an axon, and we have the myelin sheath. Now, in between the myelin sheath, the spaces here, we call it the node of Ranvier. Okay, the nodes of Ranvier, or the node of Ranvier. So you see, we have one node of Ranvier here, we have one node of Ranvier here, we have another node of Ranvier here, and then we have the myelin sheath. So there's the myelin sheath. Within the myelin sheath, we have the squam cells. Is that okay? Great. Now, and I also mentioned that the junction between the soma and then the axon, the very border between the soma and the axon, or the junction between the soma and the axon. We call it the axon hillock. The axon hillock. The axon hillock. Okay, and this is where action potential begin. Keep it in your mind, though. You see, when I'm teaching, I emphasize on certain important things in anatomy. This is where the action potential, or what we call signal conduction, this is where it begins. So the signal or the impulse is received by the dendrites. But then this is where action potential begins. Is that okay? The axon hillock. It initiates. This is where action potential is initiated, of nerve impulses are initiated. So in anatomy, if we ask you that on a typical neuron, where do action potentials usually begin, or where are they usually initiated? Then your answer that you are going to look out for is the axon hillock, which is the junction between the soma and then the axon. Is that okay? The junction between the soma and then the axon. Okay, great. So we have the dendrite, we have the soma or the cell body. Within the soma, we have the nucleus. Then we have the axon. And then we also, the junction between the soma and the axon, we have the axon hillock. Then we have some neurons with myelin sheaths. Some neurons do not have myelin sheaths. Neurons that have myelin sheaths, impulses are transmitted faster in those neurons as compared to neurons without the myelin sheaths. And within myelin sheath, we have a Schwann cell. Okay, and then at the very end of the axon, we have the axon terminals or nerve endings or synaptic knobs. Synaptic knobs. Is that okay? Great. So basically, these are the typical structures of or part of a typical neuron. Now, we also discuss what we call neuroglia in anatomy and physiology, neuroglia in nervous system. And we mentioned that when we say a neuroglia, another name for neuroglia is glia cells. Okay, another name for neuroglia is what we call glia cells. Now, when we say glia cells is a non-neuronal cell that acts as caretakers. If you remember, I told you that glia cells, they are like police officers and military men in the nervous system. So they serve as caretakers, or they serve as protectors, or they provide support for neurons. So when you hear of glia cells or neuroglia, it's the same. It's the same word. Either glia cell or neuroglia is the same thing. So either you call it a glia cell or neuroglia. But when we say a neuroglia simply means a non-neuronal cell. It's not a neuron. Neuroglia are not neurons. That's why we are calling them non-neuronal cells, meaning they are not neurons. And that's why the name is non-neuronal cell. So these are cells that act as caretakers. They are like caretakers, more like they are protectors, or you can say they are support crews for neurons. They support neurons. Is that okay? Good. Now remember, there are about 1 trillion neurons in the nervous system. We have a lot, okay, of neurons in the nervous system. But you know, even though we have a lot of neurons in the nervous system, 10 times as many neurons in your body as there are stars in galaxy, yet glia cells make up over half of the brain's volume and outnumber neurons by up to 50:1. Okay, so here, what I'm telling you is that even though we have a lot of neurons, but glia cells, their numbers outweighs or outnumber neurons at a ratio of 50:1, meaning if you take 50 glia cells in the body, there is just one neuron. Is that so? Glia cells are a lot, and it's very important because you see, neurons have to be protected. Yes, neurons have to be protected. Glia cells are like protective cells for your nervous system, and that tells you how special and how sensitive the nervous system is in terms of its protective, mechanisms. So the glia cells that protect the neurons, they are even more. They are 50 times more than the neurons. Is that okay? Yes, and that tells you how special the nervous system system is. Okay, great. Yeah, so we spoke about glial cells, and we even discussed we have types of glial cells in the central nervous system, and we have types of glial cells in the peripheral nervous system. Okay, so the glial cells, we have some inside the central nervous system. When we say central nervous system, we are looking at the brain and the spinal cord. When we say peripheral nervous system, we are looking at the nerves and the ganglia. So we have some specialized glial cells in the nervous system, and we also have glial cells in the peripheral nervous system. Now, in the central nervous system, the glial cells there are 4. So the oligodendrocytes, the astrocytes, ependymal cells, and then the microglia. So these are 4. So I can ask you that how many glial cells do we have, or neoglial cells do we have in the nervous system? Sorry, the central nervous system. So we have the oligodendrocytes, the astrocytes, ependymal cells, and then the microglia. These are the glial cells in the central nervous system. Now, when you come to peripheral nervous system, the glial cells there, we have the squam cells, we have the satellite cells. They are 2. So overall, we have about 6 neoglial cells in the in the nervous system in general. Okay, so we have 4 glial cells in the central nervous system, 2 glial cells in the peripheral nervous system. So we can ask you a question to find out whether you know the glial cells in the peripheral nervous system, and again to also find out whether you know the glial cells in the central nervous system. Okay, so we can ask you a question like, okay, one of the following are glial cells. One of the following are glial cells in the central nervous system, except. So you see A, oligodendrocytes, B, astrocytes, C, microglia, and D, squam cells. Then your answer will be squam cells. Is that okay? Another question we can ask you is that, okay, which of the following is a glial cell in the peripheral nervous system? A, satellite cell. B, ependymal cells. C, astrocytes. And D, microglia cells. Your answer here would be A, squam cells. Is that okay? Again, we can also ask you questions relating to the functions of the glial cells. Okay, yes, so when we ask you a question relating to the function of a glial cell, now, for example, if you take oligodendrocytes, if you remember, I told you that the brain is made up of neurons. The brain is made up of neurons. The brain that we see in diagrams now is neurons that are inside. The neurons that I showed you is the neurons that forms the brain. The spinal cord is neurons that forms the spinal cord. It's nerves that forms the spinal cord. Is that okay? Now, inside the brain, some of the neurons, in fact, most of the neurons inside the brain are having myelin sheaths. Because you see, in the brain, impulse conduction have to be very fast. So most neurons in the brain, in fact, I would say all neurons in the brain, they are made up of what? They have myelin sheaths on their axons. Now, the question is that the myelin sheaths on the axons of neurons in the brain, what forms those myelin sheaths? Is this cell we call oligodendrocyte. So when we ask you what is the functions of oligodendrocyte, they form myelin sheaths for the axons of neurons in the central nervous system. What are the functions of oligodendrocyte? They form the myelin sheaths for the axons of neurons in the central nervous system. Okay, now how about astrocytes? There is something unique about astrocytes. They are the most abundant neoglial cell, and it's something unique to us in anatomy that you have to know. It's an examinable question. Which neoglial is the most abundant? Is the astrocyte. It's something you should be expecting. Okay, which of the neoglial cells are the most abundant? And the answer to that is the astrocytes. Is that okay? Now, what's the function of the astrocyte? The astrocytes, they help to form the blood-brain barrier. Is that okay? They form the blood-brain barrier. Okay, and then they are the most abundant. Ependymal cells, what do? They line the brain ventricles and then the spinal cord canal. What do? They produce and help to circulate cerebrospinal fluid. So we can ask you, which of the glial cells helps to produce and helps with the circulation of cerebrospinal fluid in the central nervous system? The answer is the ependymal cells. Which of the glial cells or which type of glial cell helps with the circulation of cerebrospinal fluid in the central nervous system? The answer is ependymal cells. Which of the neoglial cells fills or lines the brain ventricles and spinal cord canal? The answer is ependymal cells. Okay, now we also spoke about the microglia. Microglia, they help to remove debris and pathogens and damaged cells from the central nervous system. The microglia cells, they help to remove debris. They help to remove what? Pathogens. They also help to remove damaged particles from the central nervous system. Okay, now let's come to the glial cells in the peripheral nervous system. The glial cells in the peripheral nervous system. Now, for the glial cells in the peripheral nervous system, we have two. We have the Schwann cells and we have the satellite cells. We have the Schwann cells and we have the satellite cells. Okay, so the Schwann cells, we have the Schwann cells. You remember I told you that oligodendrocytes, what do? They form the myelin sheets around axons of neurons in the brain. They form myelin sheets around axons of neurons in the central nervous system. They form myelin sheets around the axons of neurons in the central nervous system. Now, Schwann cells, if you come to peripheral nervous system and a particular neuron is having a myelin sheet, the Schwann cells also form myelin sheets around the axons of neurons in peripheral nervous system. So the Schwann cells also form myelin sheets around the axons of neurons in the peripheral nervous system. Okay, then we have the satellite cells. The satellite cells, they surround neuron cell bodies in ganglia. They surround cell bodies of neurons. They surround cell bodies of neurons, satellite cells. Satellite cells. Okay, so we discussed all these things. So basically, we are just revising some of the few things we discussed in our previous meeting. Now, when we say saltatory conduction, who remembers what it means? When we say saltatory conduction, what does it mean? Yes, when we say saltatory conduction, who remember? How did we say about saltatory conduction? Yes. Who remember? We discussed saltatory conduction. What does it mean? All right. So with saltatory conduction, we mentioned that if a neuron is having a myelin sheet, the membrane potentials only takes place at the node of Reynolds. If a neuron is having a myelin sheet, so the membrane potential only takes place at the node of Reynolds. Then when the impulse is generated, it jams where the myelin sheet is. Then the membrane potential will take place at the node of Reynolds again. Then for the impulse to be transmitted, it will jump the myelin sheet to the next node of Reynolds. So for neurons that have the myelin sheet, okay, good. The membrane potential only takes place at the node of Reynolds. Then the action potential jams the myelin sheet to the next node of Reynolds for the membrane potential to take place again. Then the action potential will now jump. Yes. Someone wanted to talk? Okay. So for neurons that have the myelin sheaths, the membrane potential only takes place at the node of Rehnwein. Okay, so that kind of action potential where the action potential or the signal have to always jump the myelin sheath and the membrane potential will take place again at the node of Rehnwein, then the impulse will jump the myelin sheath again. That is what we call saltatory conduction. Okay, so the action potential only occurs, the impulse transmission only occurs at the node of Rehnwein. Is that okay? That kind of impulse transmission is what we call saltatory conduction, and it only happens on neurons that are having the myelin sheath. Okay, it only happens on neurons that are having the myelin sheath. Okay, so quickly, let's go straight to what we are supposed to. And then we also look at reflex action. Okay, we also look at reflex action. Yes, so reflex action is also another very important area or aspect of the nervous system anatomy you have to know. And this reflex action is very important anatomically because you see, it helps to protect your body from danger. It helps us to regulate internal body functions like blinking of your eye, coughing, and all these physiological activities. They use reflex actions. Okay, so reflex action is more like an automatic, rapid, involuntary response to stimulus. Okay, and it's relevant to protect your body from harm. Okay, typically, a typical reflex action happens usually between 0.1 to 0.3 seconds. Is that okay? And that is also very important. It's something that you have to know. Okay, now another thing we can ask you in anatomy, a question on in anatomy, is that at least know all the structures that are involved in a reflex action. Okay, so for the structures that are involved, you see we have the stimulus, which could be a hot object. You can have a physical stimulus, you can have a chemical stimulus, you can have a thermal stimulus. Okay, good. So we have the stimulus, we have the sensory neuron. So you see, so this is a pin that is stimulating a part of the body, the skin. So the receptors in the skin will pick the stimulus. The stimulus will be transmitted along the sensory neuron to the relay neuron or the interneuron in the spinal cord. This one does not go to the brain. It happens, reflex action usually happens at the level of the spinal cord. Okay, so the sensory receptors in the skin or the part of the body will pick the information, or it will pick the signal. The signal will be transmitted along the sensory neuron, and then the sensory neuron will relay it to the relay neuron. The relay neuron receives the information, then relays or transmits the information to the motor neuron. Then the motor neuron takes the response from the relay neuron to the parts of the body to take an action. So if, for example, it was your hand that someone was pricking with a pin, let's say you are walking in the midst of class, someone prick your hand with a pin, reflex action will take place. So the reflex action, the receptors in your skin will pick the signal, send the information to your spinal cord through your sensory neuron. Okay. Now the sensory neuron will now relay the information to the relay neuron, or how we call the interneuron. Then the interneuron will now integrate and process the information and sense the situation. Then it will send a response back to the part of the body through the motor neuron. Let's go over it for the last time. So when we ask you to describe reflex action, for example, if someone touches a hot object, that hot object is a stimulus, which can stimulate the receptors at the area we are talking about. Then it will send a signal to the sensory neuron. And the sensory neuron relays the signal to the relay neuron or the interneuron. Now, within the interneuron, that is where processing of information takes place. Then the information is processed, integrated. Okay, then response to be sent back to the motor neuron. Okay, so that the motor neuron takes the response back to that part of your body for you to take an action. Either you are raising your hands, either you are changing your position, you are removing your leg from that hot object. That is what's happening. So in a typical reflex action, you see we have these structures. We have the stimulus, we have the receptor, we have the sensory neuron, we have the relay neuron, we have the motor neuron, we have the effector, which can be a muscle or even a gland. So we have the stimulus, we have the receptors. The receptors are connected to the sensory neuron. From the sensory neuron, we have the relay neuron. From the relay neuron, we have the motor neuron. And from the motor neuron to the effector. For the last time, we have the stimulus. From the stimulus, we have the receptors, which are usually in the skin or the part of the body. The receptors are connected to the sensory neuron. The sensory neuron relays the information to the interneuron. Interneuron processes and integrates the information. Response is sent back through the motor neuron to the effector. The effector is the part of the body that will take the action. Is that okay? And this is what happens. This is the structures involved in a typical reflex action. So if we ask you in exams that list all the structures that are involved in a typical reflex action, is simple. So the stimulus, the sensory neuron, the interneuron, the motor neuron, and then the effector. Is that okay? Then you list these structures. We can ask you, what's the importance of a reflex action? A reflex action helps to protect your body from danger. A reflex action also play a role in regulating internal body functions like blinking of your eye and then coughing. Is that okay? Because it's not all situations that happen in your body that we need information to go to the brain and back. Because sometimes you need a very rapid response, and your body may not be patient enough to wait for information to go to your brain. That's why reflex actions involve the relay neuron at the level of the spinal cord and not necessarily going to up to the brain. Okay, so basically that is it for reflex action. Okay, so at least know the structures involved, the stimulus, the receptors. The receptors are connected to the sensory neuron connected to the relay neuron connected to the motor neuron connected to the effector. Okay, that is fine. So basically that is it for reflex action. That is it for reflex action. So you see here, this person is touching a hot object. Once you touch the hot object, sensory receptors will pick up the signal. Okay, sensory receptors will pick up the signal. So the moment she touches the hot object, sensory receptors in the skin will pick up the signal. Once they pick up the signal, they release it to the sensory neuron. Sensory neuron takes it to the spinal cord, relays it to the relay neuron. The relay neuron also relays the information to the motor neuron. The motor neuron sends information to the muscle or the effector so that an action will be taken. Either the muscle will contract to pull your hands away, or contract to pull your legs away, or that part of your body involved in the danger away. Okay, so that is what you call reflex action. Reflex action. Who can describe reflex action? Yes. Yes, who can describe reflex action? In a typical reflex action, what happens?
By the sense.
Yes.
The
Yes, you
stimulus
want to
is
try?
detected by the
Okay, go
yes,
ahead.
sir.
Yes, go ahead. Yes, go ahead. We are
First,
listening.
the
Go ahead.
stimulus is detected by the sensory receptors,
Excellent. Good. Excellent.
and
Then the receptors will send the information
then
to.
the sensory nerve,
Yes, the sensory neurons. Yes. Yes, that is right. Continue.
and the sensory neurons will also carry
They will carry the impulse
impulse
to
to the interneurons.
interneuron. Excellent. Yes.
The
Then the
interneurons.
interneurons would also. Yes. Then the interneurons would also
Interneurons
do
will
what?
then reprocess,
Come.
reprocess the information
Mm-hmm.
back to the motor
Okay. Okay, that's fine. Then from the motor neuron to
to the affector.
good the effectors. Okay, once the information response get to the effectors, then an action will be taken to solve the problem. That is excellent. That's great. You've done well. Okay.